Hello, animal science class. I got a little review for us. We've been talking about cell division. Obviously, we went over mitosis first, which is the replication of one cell to another cell, which are identical. We started talking about meiosis, which is way more exciting. We talked about the difference and the differentiation that we get in our meiosis process, which happens in the gonads, and it is the sex cells division. Awfully exciting when we start talking about reproduction. Meiosis is very interesting because we have to get to a haploid number. All other cells are in a diploid number, and we create another diploid number in mitosis. But when we get to meiosis, we go through this division process twice and end up with haploid numbers that we call gametes, which happens to be the sperm and the ovum. So they need to be haploid number because when the sperm and the ovum meet, we create a new diploid cell, which then divides and divides and divides and turns into our animal or person, whichever it may be. This is the process, just like in mitosis. Meiosis starts out with prophase, where we get duplication of our chromosomes, but in this stage we have something called crossing over. This goes on with Mendel's genetics, where it talks about segregation and independent assortment, and through this crossing over process we go through the rest of the phases, and we have differences and different genes on our chromosomes, which you can see noted by the colors on our chromosomes as we go to metaphase and anaphase and then on to telophase. And this is all in meiosis 1. We go to meiosis 2, which just takes those cells that we just created and we divide them again. We have to divide them again because, yes, they need to be in a haploid number. As you can see, our end result, we have 4 haploid cells that are all different. This happens, as we know, with crossing over, prophase 1, but we can see here how those chromosomes then split. So as we get the genes to cross over and share on different tails of these chromosomes, then we pull them apart into different new gametes. We have variation. If we look, we can see this is obviously spermatogenesis. And it's the process of the male going through and producing sperm cells. This happens in the testes. It started by the Sertoli cell in the testes, which we know is not active until puberty. So we have no sperm cells until puberty. And then when the Sertoli cell starts producing these spermatogoniums, they go through the process of meiosis 1, meiosis 2, and we have 4 functional sperm cells that all are different in terms of their genetic codes. If we look at the female side, we call this oogenesis. The difference is the female is born with all of those primary oogoniums that they're going to have through their life. They just are not recruited and developed until puberty, and we get the brain involved, and GnRH starts to be released to trigger that ovary to recruit the next follicle. But they go through the same process, meiosis 1, meiosis 2, and we end up with only 1 functional ovum and 3 polar bodies. This is very important that we remember. It is a big difference between the male and the female, but it has to be this way because there is only 1 of those 4 that has enough cytoplasm to maintain a pregnancy until it gets to the uterus and can implant and then feed itself from the nourishment of the host or the mother. If we were to divide the cytoplasm among all 4, meaning the 3 polar bodies got an equal amount, none of these pregnancies, when it got fertilized by the sperm cell, would have enough energy and nutrients to survive those 8 days or so until it hatches and has to live off the uterus, and so it would be non-functional. So what we end up with is 1 functional ovum and 3 polar bodies that have the same genetic material but not enough cytoplasm to maintain a pregnancy. Here's a picture of our ovum being attacked by sperm cells, and we know these sperm cells are trying to get through the cumulus ophorus, through the corona radiata, and then through the zona pellucida to create a fertilized embryo that can then slide on down the oviduct to the uterus, where it will implant and grow to be a healthy, happy young calf. This slide just giving us a little bit more on the crossing over. It's very important that we understand that the difference and the genetic variation happens at this stage, and we see those genes splitting up. I have some practice problems for us to work on here in a second. You can see again how that crossing over moved our genes from one spot to another, so we have genetic diversity. We've looked at this slide before. This is just a couple of homologous chromosomes, which means they're alike, and it's showing a pair of alleles. And alleles are just those genes that occupy similar locations on homologous chromosomes, which might be a gene for eye color or hair color. Our example, we're going to use cattle, and we're going to use genes for black and red, which we know go straight on the dominant recessive situation. You can see I have put some A's and B's on our chart where we can see genes that match up for different alleles. So we know that on those same spots of our chromosomes, we are going to match. In our higher level species, we know that Mendelian genetics doesn't explain everything. We know that there are multiple alleles for traits, and that there are still situations where we have codominance, incomplete dominance, which we'll talk about later when we talk about roan color. It's not straight dominance, or we would be red or white. So we will explain some of those. But for now, let's think about Mendel and his straight dominant recessive type situation. I have a Punnett square here at the bottom of the screen, and this Punnett square is going to help us determine how many different types of colors we can get, or phenotypes in our offspring. I have labeled this as large A, which is dominant, small a, which is recessive. So in the next example, we're going to use A as black and small a as red, because we know that red in our cattle genes are recessive. So when we are trying to figure out the number of gametes, if this is the whole genome for our cow, just one color gene, which we know is not true, there are thousands and thousands of them, we have to get that into a haploid number, right? So haploid means one of these will be sent on the chromosome and one will not, right? So in our sire, when we are making our sperm cell, we know that he has a choice of creating a large A, which goes in this block, or a small a, which goes in this block. Those are our only two choices of gametes. On our dam, she has the exact same genotype, which means that she has a large A for black and a small a for red. And when we set up her gametes, she also has two, a large A which goes here and a recessive A that goes there. When we start filling out this chart, all we do is simply match them up. So if you follow the arrows in step 1, we have our two little A's that go in that block, which means that one will be red. As we move over, this A comes down, small a goes over. Remember, when we write these, always put the dominant before the recessive if there is a heterozygous pair, meaning one dominant, one recessive. As you roll across here till we get to step 4, all it is filling in the boxes, so hopefully that makes sense. When we look at this, we can then determine what percentage or what chance we have of getting certain phenotypes, and the phenotype is what it looks like. Genotype is actual gene makeup. Phenotype is what our animal is going to express. In this case, we would have 3/4 of them being black and 1/4 being red. So when we make this, the possible gametes for the sire are, in this case, we have a gamete for the large A, and we should have a gamete for the small a. When we do the dam in this case, we only have one gamete choice of the large A. So our Punnett square won't be a square. It'll be a rectangle in this case. When we look at this, genotype is the gene combination. So these two, the sire and dam, both express a black hair coat. But genotype-wise, they are different. So they're the same phenotype, but different genotype. Obviously, we can see heterozygous here, homozygous here. And so the genotype is what expressed. Homozygous means alike, heterozygous is different. Dominant is always written down before recessive, and the dominant is always expressed. The only time a recessive is expressed, yep, you guessed it, is when it's homozygous recessive. Here's our phenotype on that genetic cross. We cross this sire with this dam. So this is black, meaning a heterozygous pair is black. Our homozygous pair is black, and the only time we get red is homozygous recessive. So as we start thinking about and we start making these crosses, there are two different ways. So make sure that you read any worksheets to find out whether or not we want to make a genotypic variation or a phenotypic variation on our ratios when we do it. In this case of the one we just did, you would have 50% genotypic that were homozygous for black and 50% that were heterozygous for black. But on phenotype, we are 100% black. Hopefully that makes sense. I'll send you a worksheet and you guys can do some practice problems. And I'm hoping that you have a great and wonderful weekend, and I'll see you on Monday.